All right, everyone. We're going to go ahead and get started. So if we can get your attention, that would be great. Yeah, so we have now the second of the two short communication sessions. I think you'll agree with me that yesterday's session was really fantastic. A lot of exciting work going on. And so today it promises to be just the same. So I'll just quickly introduce myself. My name is Flaviano Giorgini. I'm a professor of neurogenetics at the University of Leicester in the UK, and I'm also one of the science directors at EHDN.
And I'm Davina Hensman. I'm a consultant neurologist and a researcher in Huntington's disease based at UCL Huntington's Disease Center.
All right, and we'll go ahead and get started since I see our first speakers are ready to go. So we have Ella Matthews, who's a research scientist from the University of Washington in the lab of Jeff Carroll. And she's going to be telling us about blocking Huntington's transcription protects against somatic instability. On to you.
Thanks to all the organizers for inviting me. I really appreciate the opportunity to be here. Also, I want to say that this is just a very condensed summary of all the data that we've gathered on this project. So if you want to see all the different Huntington lowering modalities we looked at, it's poster A11. So we know that somatic instability is a key driver of Huntington's disease onset. The more instability someone has, the earlier symptoms will manifest. And currently, the impact of Huntington lowering treatments on instability is unclear, even though Huntington lowering treatments are pretty much the primary method of therapeutic treatment right now. Some treatments might decrease instability, but for many, we don't know what their effects will be. So because of this, we wanted to look at the interplay between several different Huntington lowering modalities in mice, and we wanted to quantify their impact on instability. So the first thing that brought this to our attention was when we used an ASO targeting exon 36 of Huntington in Q111 mice. And we already know this ASO is successful at reducing Huntington protein levels. So this trace here is how we measure instability. It's generated from a CAG spanning PCR, and it shows fragment length on the x-axis and the quantity of each length on the y-axis. So the left-hand peak is the inherited allele, which in this case is Q111, and the smaller peak on the right is the expansion, which increases over the lifespan of the mouse. So in this saline-treated mouse, the lengths are skewed to the right compared to this ASO-treated mouse, which shows that ASO treatment may be able to reduce instability. And we didn't initially expect this ASO to affect instability at all, because it's supposed to act on mature mRNA. But we think it might also bind to pre-mRNA in the nucleus and interrupt transcription, which then lowers instability. So to test this, we looked at pre-mRNA in 3 different locations along the Huntington transcript, and we found that all 3 of these locations are decreased in ASO-treated mice. So to further investigate the effect of Huntington lowering on instability, we used a ZFP developed by Sangamo, which is comprised of 2 different parts, a DNA binding domain targeting CAG repeats and a DNA repressor domain. And we also used both of the parts individually in 2 different control arms. So as expected, only the combined construct was effective at reducing Huntington levels, because it can both bind CAGs and repress it. And then we looked at somatic instability. So on the left, the DNA repressor by itself doesn't have any impact on instability because it can't bind to Huntington. And then on the far right, the combined construct reduces instability by 76%. But the surprising thing here that we saw was in the middle, which is the DNA binding region only. And this reduces instability by almost 50%. So this suggests that just binding CAGs alone is enough to disrupt instability. And it shows that there are probably 2 different mechanisms at play here, both reduction of transcription and steric hindrance from binding CAGs. So because of the previous apparent impacts on transcription of Huntington lowering, we wanted to look at a way of more directly silencing Huntington's transcription. So we used a CRISPR dual guide system to delete the entire promoter of Huntington while leaving the CAG repeats untouched. This stops transcription, and it sharply reduces instability. So this suggests that transcription is necessary for instability to be able to progress at its normal rate. Because of the previous data showing that lowering Huntington lowers instability, we initially had the thought that the protein itself might be involved in the instability of its own gene. And to investigate this possibility, we wanted to see if lowering Huntington outside the nucleus would affect instability. So we used an siRNA that's only active in the cytoplasm. And it lowers Huntington just as much as the previous methods, but it leaves instability completely unaffected. So that shows us that lowering Huntington post-transcriptionally is not sufficient to reduce instability at all, and the protein is likely not involved in this process. So to recap, we know that transcription of Huntington, even without any protein production, is sufficient for instability to progress at the normal rate. Then we also learned that different methods of lowering Huntington that act via reducing transcription are effective at reducing instability, which points to a direct role of transcription in the expansion process. Then the last thing we found is that binding CAGs alone, even without Huntington lowering, is still enough to protect from instability. And I want to thank everyone that contributed to this project, especially the entire Carroll Lab.
Great job, Ella. Thank you for that. Do we have any questions? I think we have time for a question from the audience. Can't see out there very well. No? Maybe I can get started and just ask. So how do your results then, what would be the next steps in terms of implications from your results?
We're really trying to narrow down on what the exact mechanism of this is, and so we're looking at doing a few different types of chip assays to look at where the RNA polymerase is actually bound to on the transcript to try to see how lowering transcription actually affects instability.
Okay, yeah, so mechanistic studies. Oh, we have a question out there.
So do you know what actually, hi, Kura. I wanted, if you know what things actually do control transcription, and could we find ways to decrease transcription as a mechanism?
Yeah, that's a really good question. That's something that might be worth looking into. I don't know any specific ways.
Okay, well, I think then we'll thank Al again for the great talk. So next up, we have Jasmine Donaldson, who's a research associate at University College London with Sarah Tabrizi, and today she'll be telling us about FAN1 and protecting against CAG repeat expansions.
Thank you so much. I would also like to start by thanking the organisers for letting me talk about our research, which is looking at the functions of FAN1 that protect against CAG repeat expansions in HD cell systems. So as you've heard a lot of today, a number of genome-wide association studies have found multiple mismatch repair genes and proteins are associated with an altered age of onset. A number of these genes were already associated with Huntington's disease as drivers of repeat instability, but the most significant genome-wide signal was at this locus at chromosome 15, which was identified as FAN1. FAN1 was really interesting because it hadn't yet been associated with Huntington's disease, and its canonical role was in interstrand cross-link repair. But when it was first identified, a number of labs, ours and others, identified that knocking out FAN1 potentiated repeat instability both in Huntington's disease models and also models of Fragile X, so CGG repeat expansions. We and others have then since been very interested in the functions of FAM1 that protect against repeat expansions. FAM1's nuclease has been associated as being required for these expansions, but we are also really interested in the interaction that FAM1 has with a number of mismatch repair proteins that were also identified in the GWAS. So our research, or the research that I'll talk to you today about, is really interrogating the functions of FAM1. So what we first did was we used a stem cell model with 125 CAG repeats derived from a patient with Huntington's disease. This cell model shows somatic instability in culture, both as stem cells, so dividing cells, and also when we differentiate it to medium spiny neurons, which are postmitotic. What we first did was we we introduced an alanine mutation in the C-terminus of FAM1, rendering, in the nuclease domain, rendering it nuclease dead. We then performed, so FAM1 is an interstrand cross-link repair agent, so when we treat with mitomycin C, mitomycin C is an interstrand cross-link repair agent, and FAM1 should repair these interstrand cross-links. You can see that both the D960A, so the nuclease dead cells, and the FAM1 knockout cells have reduced viability as they're not able to repair these cross-links. We then performed expansion assays in iPSCs, and they're ongoing in MSNs, but effectively what we see is the expansion rates of the nuclease dead cells expand at the same rate as the FAM1 knockouts, which was what we were expecting based on previous results from other labs. So really our main focus has been really trying to characterize the interaction that FAM1 has with mismatch repair protein MLH1. So our lab and others identified a key motif at the I'm not very good at working this. Identified a key motif at the N-terminal of FAM1, which we have dubbed the SPIF motif, and this is involved in MLH1 binding. And what we did was we created a double mutation in this SPIF motif, where we mutated the tyrosine and the phenylalanine residues to alanine. And when we did this, we see a reduction in MLH1 binding in a dose-dependent manner. And when we perform expansion assays in both stem cells and medium spiny neurons, we see that this mutation increases expansion rates to rates comparable to FAM1 knockout. Now, this was both amazing but also quite interesting, because if we're honest, we weren't expecting such a big effect of these SPIF mutants. And this really led us to wonder, first, we thought, the FAM1 MLH1 interaction is clearly important, but we also wanted to check whether we're still getting a functional FAM1 protein produced with our SPIF mutants. So what we then did was we wanted to test whether we were still getting normal canonical kind of nuclease activity in our SPIF mutants. And so we performed a nuclease assay with fluorescently labeled synthetic substrates with a 2-CAG loop out. And we took FAM1-enriched cell extracts from each of our mutant lines. And what you can see is that if FAM1 endonuclease activity is working, you should get an endonuclease cut about 35 base pairs into our 50 base pair substrate. And you can see in the SPIF mutants, we're still getting this endonuclease activity, but it's completely lacking in the D960 and the FAM1 knockout. So this was brilliant. It suggested that we're still getting functional nuclease activity, but does suggest that the SPIF motif could be regulating other domains in FAM1. So to summarize, we see that FAM1 protects against repeat expansions in a HD cell system. Mutation of the FAM1 SPIF motif both reduces MLH1 binding to FAM1 and accelerates CAG repeat instability. SPIF mutants still show functional FAM1 nuclease activity. And it suggests that both the FAM1 MLH1 interaction and FAM1 nuclease activity are key in protecting against CAG repeat instability. So we're now really wondering whether the SPIF motif is involved in FAM1's recruitment to CAG repeats, and we're exploring this with both FAM1 chip-seq experiments and also looking at the wider interactome with these SPIF mutations. So with that, I'd just like to say a huge thank you both to Sarah, who is brilliant, and in particular to Claire Pimblett, who has done a huge amount of work with the SPIF mutants, and Jess and Marwa, who have been optimising the nuclease assay. So thank you.
Thank you for the excellent talk, Jasmine. Do we have any Fan One fans out there who want to ask an urgent, burning question? No. Maybe I can just ask you a quick one. So what are the ways that, that you and others are thinking about in terms of harnessing this sort of data for therapeutics or, dealing with these interactions or blocking them or something along these lines?
I think this is the key question, really. So in our lab in particular, at poster C10, Jess is looking at phosphorylation of FAM1. So phosphorylation of the serine residue in the SPIF motif inhibits the FAM1 MLH1 interaction. So if we can kind of prevent the phosphorylation using kinase inhibitors, this could represent a therapeutic opportunity. So that's something that Jess Olive is looking at. I know there's a number of research labs looking at upregulating FAM1, and we don't know how tolerated the upregulation will be. I know there's some data out there suggesting that we can almost upregulate FAM1 to a certain level and it won't have any damaging effects. But again, I think we have to be careful with how much we upregulate DNA repair genes or proteins.
Great.
Thank you.
All right, thank you for that. Let's thank Jasmine once again.
Thank you. I'd like to welcome Anat Bahat, Associate Staff Scientist at Weizmann Institute of Science. Thank you.
Thank you. So good evening, everyone. I would like to thank the organizers for giving me this great opportunity to tell you about our small molecules that can be a potential treatment for Huntington disease. So our target is the SPT4-SPT5 complex. It's a transcriptional elongation factor that interacts with RNA Pol2 in order to stabilize it during transcription elongation. It is specifically required for transcribing expanded repeat genes that are associated with neurodegenerative diseases such as Huntington disease, ALS, FTD, and SCAT36. And in all these cases, it was shown that depletion of either SPT4 or SPT5 reduces the transcription level of the mutant allele without affecting the wild-type allele. And this selective inhibition probably arises from the presence of abnormal secondary structures that are formed by these repeated sequences and challenge the elongating RNA Pol2 and render it to be much less stable in the absence of SPT4 and SPT5. So given its role, this complex is a promising target for treating repeat expansion diseases. So this drove us to look for SPT5 inhibitors, and for that purpose we targeted the interaction between SPT5 and RNA Pol2 in a high-throughput drug screen, which led to the identification of the first SPT5 Pol2 inhibitors, which we named SPIs. So while most of the SPIs are considered as general or partial SPT5 inhibitors, some are selective only for the mutant HTT gene, and they are not affecting the wild-type allele or other SPT5 activities, suggesting that the various activities of this factor can be uncoupled and selectively targeted by the SPIs. So we performed the survey catalog to this small group, and we found the two improved potent analogs, which we named the SPI24 and SPI77. And this analog indeed inhibit only the mutant HTT gene and not the wild-type gene in a mouse striatal cell line bearing 111 CG repeat, and also in HD patient-derived heterozygous cells bearing 44 CG repeat. And in BAC-HD mouse, administering these drugs by different delivery methods resulted in a significant reduction in the mutant HTT level in the brain striatum, along with improvement in HD biomarkers and HD symptoms as expected from a successful lowering of mutant HTT. In addition, pharmacokinetic analysis revealed that both SPIs can pass the blood-brain barrier. So we examined the effect of long-term subcutaneous SPI-24 treatment on the disease progression of early-stage BAC-HD mice by different behavioral tests. And the BIMWalk test showed significant aggravation over time in the slippage ratio of the non-treated group. However, this aggravation was completely abolished in the SPI-24 treated group. In addition, in the home cage locomotion, we saw a significant reduction in the involuntary movement of these treated mice. And the wheel test as well as the climbing test both showed a significant improvement in the motor skill of the treated group with performance which is approaching to that of the wild-type group. I want to mention that we did the same experiment with oral delivery of these drugs, and we got very similar results. So this prolonged treatment had no side effects or global changes in gene expression. And in conclusion, I would like to say that the SPIs demonstrated the advantage over other HTT lowering approach with respect to mutant HTT selectivity and simpler delivery methods due to BBB permeability. And this resulted in improvement in HD biomarkers and HD symptoms and the delay in the disease progression with no apparent toxicity. I just want to mention that we have preliminary data that showing that the same SPIs can reduce also other expanded repeat genes like SCAR1, 2, and others. And with that, I would like to end and thank all the great people that took part in this study, to our founders, and thank you for your attention.
Thank you for that lovely talk. Do we have any questions from the audience? Maybe I'll kick off. So I was just curious about the off-target effects, if it's something that is going to affect all repeats. At what size does it kick in, and how do we deal with off-target effects?
In Huntington, you mean?
Well, across the genome. Was it specific?
Off-target effect. Okay, so actually I moved quickly about it, but we did do a RNA-seq for the brains of treated mice after the prolonged treatment, and we saw only few genes that were upregulated, which is very small number.
Thank you.
Thank you. I had another quick question for you. So in terms of the SBIs, what's the mechanism of the inhibition? Is it just blocking the interaction with Pol2? Or is it
So actually, what it's doing, it's interfering the interaction, change the conformation between SPT5 and RNA Pol2. And since SPT5 has several functional domains, it affects only, these specific SPIs affect only a specific activity of SPT5. And we know that SPT5 is required in order to stabilize the RNA polymerase in complicated sequences like this one with many repeats. Therefore, the change in conformation led to the fact that it's much less stable and it even falls from the DNA.
Okay, great. Well, thank you very much.
Thank you.
So I would like to invite Farah Kordapour Delmani, sorry, I'm really sorry for the pronunciation of your name, to give us the next talk, and she is a research associate at the University of Glasgow. Thank you.
Okay. Hello, everyone. Thanks, organizers, for letting me to present some data today about my postdoc project on a potential therapeutic biomarker. So we know that the somatic expansion of CAG repeats in HTT gene has an important role on the onset of Huntington disease, and anyone born with more than 35 CAG will have somatic expansion over a lifetime, leads to expanded CAGs and polyvoltamine, neurodegeneration, and finally Huntington disease. We also know that the more CAG repeat, the earlier age at onset. So any drug that can suppress the somatic expansion might be beneficial for the patients. However, we need a biomarker for the drug development to detect the effect of the drug on the somatic expansion, but we don't have access to the brain cells. But we thought that if any drug is exposed to the periphery and not directly to the brain, we might be able to detect the effect of the drug on the somatic expansion in the blood. So in order to investigate that, we worked on 268 Enroll-HD patients with 47 to 69 CAG repeats. We selected the patients with larger allele because we knew that those patients have more somatic expansion. We got 2 to 6 time point samples per patient, almost 1 year time interval, totally 1,013 DNA samples to answer the question, what is the change of somatic expansion over time? We did an ultra-deep sequencing assay, and this is for 3 replicates on each sample. This shows the repeat length distribution for each sample. CAG length is on x-axis, number of reads on y-axis. The major peak is N, which is modal allele. Peaks before that are PCR slippage or PCR artifact, and after that are somatic expansions. So with this method, we are able to measure the somatic expansion for each patient at each time point. And this shows the changes of somatic expansions over time in all the patients. So age at sampling is on x-axis and somatic expansion on y-axis. Each line refers to one patient. Each dot is somatic expansion at each time point, and the line is the line of the best fit for each patient. So as you see, most of the lines are going up, showing increase of somatic expansion over lifetime. At the bottom, we have the patients with shorter alleles, 47 to 50, 52, with a slower increase of somatic expansion, but the data is more consistent there. At the top, we have the patients with larger slopes, showing that the somatic expansion is changing faster over time. However, we have more variance there. In order to estimate that how much our power is to detect the changes over 1 year, we did subsampling on all samples with almost 1 year time interval. We did subsampling for 1,000 times with the sample size you are seeing on the x-axis, and the p-value is on y-axis. So the more the value on the y-axis, our power is also more. So as you see, with the sample size of 200, we have a very high power to detect the changes of somatic expansion over 1 year in all patients with p to 10 to minus 2, and decrease of sample size cause our power also goes down. So this is the subsampling on the all samples with 47 to 69, but we decreased we repeated this subsampling on the samples with shorter allele, 47 to 50 CAG, shown in blue here. And now you see that our power is increased, and even with 150 sample size, we have the high power to detect the changes over 1 year in all patients. So the reason might be with the samples with larger allele, we have more variance, and probably some of the signals are lost by the noise there. So in conclusion, we are able to detect the annual changes of somatic expansion with this method. In general, patients with larger allele have more somatic expansion and faster changes over time, but more variance also. We had a high power to detect the changes in natural history study, but we haven't done power calculation yet with the drug and effect size. And also, we think that some technical improvement might increase the sensitivity of our test. So finally, I thank the patients and their family, Enroll-HD cohort, my PI, Professor Darren Monkton, and his group members, especially Mike Husey and Anna Gatsavas, and the funders, CHJ and Vertex. Thank you very much for your attention.
Thank you very much. Do we have any questions from the audience? I think, is there anyone? Should we?
Yeah, in the interest of time, I think we'll just go ahead and go along. But thanks again for your wonderful talk, and we'll close the session.
Thank you very much.